genome research. Dr. Majee became fellow of our academy in 2022, and the title of his talk, SKIP JAZ by SKIP31, a key event for seed maturation and desiccation tolerance.
Thank you, Dr. Mondal, for introducing me to these audiences, and I also thank the Academy for giving me this opportunity. So I am going to talk about one of our research work on SKIP31 protein. So our lab primarily works on seed. We have been trying to understand some of the spectacular features of seed. We all know the overwhelming importance of seed in terms of agriculture, but seeds are biologically very, very interesting. It offers a lot of biological questions to understand. One of the remarkable features I have shown here, so seed can stay alive for prolonged period of time. So the seed has the ability to retain germination ability or viability for very prolonged period of time. Prolonged means really prolonged. It's not about decade, it's about a millennium. So here I am showing you two unbelievable example of seed longevity. One is date palm, which is khejur, which can be viable as many as 2000 years, and still even after 2000 years seed can able to germinate and produce a successful plant. There is another example called lotus seed, ancient sacred lotus, in Bengal it's called poddo. So they can survive as many as 1300 years, and even after that they can still able to germinate and produce a successful plant. So what is the secret behind the seed longevity? So this type of seeds which can survive in a desiccated state called orthodox seeds. So because they are desiccation tolerant, they can retain their viability for very prolonged period of time. Fortunately, most of the crop plants produce this orthodox seed like rice, wheat, chickpea. They produce orthodox seed. That's why we can store them and we can use them as per our need. So that's a huge advantage to the agriculture. There is another classes of seeds called recalcitrant seeds. So they don't survive in a desiccated state for a very long period. Even in some cases they survive only for a week or only for a day. So these are some examples like oak, mango, lychee. There are lots of fruits. They produce a recalcitrant seed. That's why they don't survive for a long period. So our lab is trying to understand how does orthodox seeds stay alive for very prolonged period of time, while that recalcitrant seeds cannot survive for a very long period. What is the mechanism underlying seed survival, vigor and longevity? How can seed survive for so long period? What is the mechanism to retain their viability or germination ability for a very prolonged period of time? And can we do something? Can we changes or manipulate these processes or genes to increase the seed vigor and seed longevity or seed storage life, which is a huge advantage for the agriculture? So what is the survival strategy of seeds? That's what we wanted to answer this. So seeds adopts a very smart strategy. It's called drying without dying. So they become dry but they don't die, and that's why they retain their viability. So how they achieve this? So during seed development, from zygote to the seed, actual mature seed, that's called seed development. So this during seed development, seed reduces the water content very methodically and organically. So particularly during the seed maturation stage at the end of the seed development, here actually, so in this phase actually, so seed drastically reduces their water content. When the seed reduces their water content, so that they become dry, and there is a lot of advantage of becoming dry because they reduce the metabolic activity, so they can save their energy because there is a very less amount of metabolic activity, so there is a very less amount of ROS accumulation or toxic metabolites, and which is also, this drying also makes them resistant to pathogens. So this is one of the hallmark to stay viable for a very long period. However, when there is a drying, so it's very difficult to survive without water. So it's very difficult to stay alive in a drying conditions. So because it causes a lot of cellular damage, functional proteins damage, structural damage of the protein like DNA damage, protein damage. So there are a lot of undesired modification happens in the seed protein like isoaspartate modification, methionine sulfoxide modification. So plant adopts a very remarkable features that is called desiccation tolerance. So seeds acquire a desiccation tolerance during the seed maturation stage. In this stage, actually, so they have acquired the desiccation tolerance property, which actually allows seeds to survive for a very long period because they have to survive in a desiccated state. So this mechanism includes protection mechanisms and repair mechanisms. So our lab is trying to understand the repair mechanisms and protection mechanisms of the seed. So this protection mechanism includes a lot of sugars, RFOs, LIA proteins, chaperone protein, HSPs. They actually protect all the protein which undergoes damage in a desiccated state in the seed. And there is a strong repair mechanism in the seed, which includes the protein repairing enzyme like PIMT, MSR. They are called protein repairing enzymes. They actually repair this undesired modification and make them functional in the next. So this is achieved during the seed maturation stage. which is actually coordinated by the ABA hormone. So ABA hormone is a very common hormone in the plant. They actually play a very important role in various seed dormancy or seed maturation, seed viability. So this ABITF actually plays a very important role to induce this protection mechanism, repair mechanisms in the seed. So our lab is extensively work on protein damage and repair seed. We have identified a very anti-aging enzyme called protein A lysoaspartyl methyltransferase, which is highly abundant in the dry seed. So what it does, we have shown that this is a key enzyme for preserving seed vigor and longevity. So what it does, during seed aging or during seed storage, the aspartate residues and the asparagine residues of any protein can spontaneously modify to isoaspartate. So this is spontaneous modification. It is induces during the dehydration stage of the seed or ROS accumulation of the seed. So when the isoaspartate is formed, it disrupts the protein original structures, which actually inhibits the biological function because of the isoaspartate modification of the protein. While the seeds have a lot of PMT enzymes, so this PMT enzyme can recognize these isoaspartate residues and convert them back to the aspartate residues. So the damaged protein become normal protein again, so which actually restores its original function, which has been disrupted due to the isoaspartate accumulation. So this PMT actually reestablishes the protein function, which is actually required for the improved seed vigor and viability. So we have extensively worked on this protein, and we have found these are the seed protein like catalase, ascorbic peroxidase, helicase, Lia protein, HSPs, lot of transcription factors. They undergo this modification and they lose their functions, while PMT can recognize this isoaspartate and make them functional again. But today I am going to talk about another protein called SKIP31, how this SKIP31 modulates ABA signaling pathway in the seed and how it improves the seed vigor and viability. So during our textbook, we all know that ABA signaling is activated or ABA hormone is increased during seed dormancy. But while germination, this ABA activation or ABA signaling should be repressed. So how seed actually attains this ABA signaling during phase transition from seed maturation to seed germination? While because in this stage ABA signaling should be activated, while in this stage the ABA signaling should be repressed. So how seed actually does this fine tuning job? So today I will talk about the SKIP31 protein, how they regulate ABA signaling pathway. So Vishal actually did this work. So first I introduce what is the SKIP31 protein. So SKIP31 is basically SKP1 interacting partner 31 is a nuclear localized F-box protein. So SKIP31 is basically an F-box protein, and F-box protein is a component of E3-SCF ligase complex, which is a part of the ubiquitin 26 proteasome pathway. So this proteasome pathway plays a very important role. It's a protein degradation pathway. It selectively degrades protein in a cells. So what it does, this F-box protein actually identifies this target protein, and this target protein is to be degraded through the 26 proteasome pathway. So first we wanted to know whether this F-box protein is a real F-box protein, as it is a part of E3-SCF ligase complex. We found that SKIP31 interacts with the most of the ASK protein. ASK protein is basically a Arabidopsis escapiloac protein, that's why it's called ASK protein. So SKIP31 interacts with the ASK protein, and it's a nuclear localized F-box protein. So next, just to understand the function of any gene or any protein, you must analyze the function, where it expresses and when it expresses. So we found out that this SKIP31 protein is highly expressed in the seed. Here you can see as compared to the other organs, and through promoter gas fusion analysis we can see that this is highly expressed in the embryo of the seed. To understand better, we actually check the expression pattern during the seed maturation or seed development stage. Here you can see in the late phase of seed development or seed maturation stage, the SKIP31 protein is highly expressed or upregulated in the late maturation stage, while this expression goes down once you imbibe the seed or once the seed proceeds for germination. That means SKIP31 may play a role during the seed maturation stage, but not during the seed germination stage. And it's also induced by the ABA, because as I told you that ABA hormone plays a very important role for seed maturation, seed dormancy. So it's also induced by the ABA hormone, and it's also induced by the dehydration. So that makes sense, because in a dry mature seed it has a lot of ABA, and it's a dehydration stress conditions. So here you can conclude that SKIP31 is expressed predominantly in the seeds and during seed maturation stage. So now to understand the function of this protein or this gene, we need to analyze the mutant. So what is the phenotype of the mutants? So when we procure the mutants from ABRC stock centers, this is in Arabidopsis plant, which is the model organisms in plant system. So here you can see we found three distinct abnormal phenotype. One is the defects in the seed morphology. So this is the mutant you can see. This is the wild type plant or normal seed. Here you can see there is a shrunken seeds, and it shows the reduced germination, significantly reduced germination, and it also significantly reduced viability. So the viability is compromised, germination is compromised, and there is a defect in the morphology. So that means SKIP31 may play a role to regulate this viability and seed germinations as well as the morphology or the development of the seed. We have confirmed this through the complementation assay, seed specific overexpression lines and RNAi lines. And when we express SKIP31 ectopically, then you can see this mutant phenotype is restored. So this is the mutant. When we express them in this mutant background, you will see the restore the typical seed morphology, seed germination and viability like wild type. That means SKIP31 plays an important role in this particular traits. We have confirmed again by generating RNAi lines as well as seed specific overexpression lines. We specifically express seed in this SKIP31 protein in a seed specific manner, and we found that there is improved seed vigor and improved viability. So overall, all this genetic experiment, we can conclude that SKIP31 play a very positive role for seed maturation and seed vigor and viability. So now there could be two possibilities. One is either is due to the defects in the embryo, seed development or seed maturation, or due to the defects upon storage, because we have analyzed all the germination and viability after storing. So what is the main cause of this defect? So we have actually analyzed germination, desiccation tolerance, phenotypic analysis throughout the seed development. This is the different stage of seed development. Here you can see this is the early stage of seed development. This is the S4, S5, S6, S7, and we found there is no defect in the early stage of seed development. So that means everything is normal like wild type. When you compare the mutant and the wild type background, so there is no defect, but the defect starts from the late seed maturation like S7 stage. Here you can see that there is abnormal seed phenotype, there is a differences in the germination as well as viability. So premature browning, shrunken seed and lower germination potential during the late seed maturation, not the early seed development stage. So till that there is no problem at all. So now to understand the actual function of the SKIP31 protein, as I told you that what could be the possible reason, because as I told you that SKIP31 is basically an F-box protein. So if you can identify the target protein which is degraded through this pathway, can actually explain the phenotype. So to find out the interactive partner of the SKIP31 protein, we did a H2 library screening, and we found a very interesting protein as an interactive partner, JAZ, jasmonic acid gene domain protein. But this is very surprising because JAZ has nothing to do with the ABA hormone signaling so far. So what it does, so JAZ actually now introduce the JAZ, the interactive partner of the SKIP31 protein, or in other words, SKIP31 target this JAZ protein. So JAZ is basically a repressor protein of JAZ signaling pathways. JAZ is another hormone which is mostly involved in the insect resistance and biotic stress responses in the plant. So what it does actually, so JAZ actually interact with the MYC2 protein, which actually is required for JA responsive genes expression, but this JAZ protein forms a complex with the MYC2 and repress the expression of the JA responsive genes. So it's basically a repressor of the JA signaling pathway. But another interesting observation was that COI1 is an F-box protein which is already target JAZ protein. So this is already known. This is very popular phenomenon in JA signaling pathway. So this F-box protein COI1 already targets JAZ protein and degrades JAZ protein, and then allow MYC2 to do their job, and J responsive genes are undergoes or expressed. So if this JAZ protein is already targeted by the COI1 protein, another F-box protein, what does it do? What does our SKIP31 do? Because this is already targeted by the COI1 protein, and this interaction is J isolation dependent, because this interaction requires J isolation hormones. So then we wanted to know whether our interaction, the JAZ protein and SKIP31 interaction, requires J isolations or not. So the next question, we wanted to know that whether interaction of SKIP31 with JAZ is coronation dependent or independent. So to know this, we actually did a Y2H analysis again in presence and absence of coronatin or J isoleucine. Here you can see that our interaction doesn't matter whether you apply coronatin or jasmonate isoleucine in the media or not, because we have supplemented media with this particular molecules. And we found that unlike COI1, because as I told you that COI1 requires coronatin molecule to interact. Here you can see if you don't use coronatin or jasmonic isoleucine, there is no interaction. However, in this case, you will see the interaction. So this interaction doesn't require, or you can say it's independent to the J isoleucine. And we have confirmed this interaction through biopsy and pull down assay. So then we again check, because we now know the interaction. So whether it actually leads to the degradation of this JAZ protein or not. We have done several experiments. One is in vitro ubiquitination assay. Here you can see that in presence of SKIP31, JAZ undergoes ubiquitination, and this ubiquitination is essential for protein degradation, because this complex recognize this ubiquitinated protein only. So what we can conclude here, that SKIP31 mediates ubiquitination of JAZ proteins and their degradation via the 26S proteasome pathway, that we have confirmed through the cell-free degradation assay. Here you can see this protein is degraded, but when there is MG132, which is a proteasome inhibitor, then this protein becomes stable. So in this experiment, we can say that SKIP31 mediates JAZ protein and their degradation through the 26S proteasome pathway. So now we got two conclusions. One is SKIP31 positively regulates seed maturation, desiccation tolerance, and seed vigor through our genetic studies. Another way, we can find that this can mediate JAZ degradation, but whether these two phenomena is interlinked or not, that we still don't know. So we wanted to know whether this SKIP31 positively regulates seed maturation, desiccation tolerance, seed vigor by mediating JAZ degradation or not. So this is another phenomenon, this is another phenomenon. So now I wanted to know whether this seed maturation, desiccation tolerance, or seed vigor is due to the degradation of the JAZ protein or not. So to confirm this, actually we have analyzed lot of mutants again. So this, if this is a positive regulator, then this should be the negative regulator, because it targets this protein and it degrades this protein. So if this overexpression lines is positively regulates seed maturation, desiccation tolerance, then this is the negative regulator of this one. So this overexpression line should behave like this SKIP31 mutant lines, because they act antagonistically to each other, and JAZ6 or knockout lines should behave like a SKIP31 overexpression lines. So that's what we did actually. So we found that these JAZ delta JAZ overexpression lines mimic the phenotype of SKIP31 RNA lines. Here you can see. So this is the overexpression lines 1, 6, and 11. There is a abnormal phenotype. There is a reduction in the seed germination, reduction in the seed viability. There is a reduction in the seed germination. So this defects. So this JAZ6 actually mimics the phenotype of SKIP31 and RNA line. So how JAZ protein negatively modulates seed maturation, seed vigor, and viability. So meanwhile, there is another report came that this JAZ actually acts as a repressor of the ABA signaling pathway. Earlier it was known as acts as a repressor of the J signaling pathway. But now we know that JAZ protein is actually interact with the ABI13 and ABI5, which is the master regulator of ABA signaling pathway, and they actually repress the ABA responsive gene expression. So then it explains our phenotype actually. So the JAZ protein interact to ABI-TFs and possibly repress their trans activation activity. So now the question is, are ABA responsive genes down-regulated in the SKIP31 RNA and this overexpression lines? So we have extensively analyzed the ABA responsive genes in this overexpression lines and mutant lines, and here you can see that this all these ABA responsive genes are down-regulated in SKIP31 RNA mutants and these overexpression lines, and up-regulated in the SKIP31 overexpression and JAZ mutants. So now we conclude that the defect in seed maturation in these mutant lines, SKIP31 RNA mutant lines, or the JAZ overexpression line is due to the compromised ABA signaling and downstream gene expression. So that explains our phenotype actually. We have also analyzed lot of things, like because ABA hormone regulates seed reserve accumulation, protective metabolites, ABA content, which actually required for the desiccation tolerance and seed vigor. But when there is in this mutant background, all are compromised. So there is a poor seed desiccation tolerance and seed vigor. While in the overexpression lines, these are improved, because in this scenario, activation of ABA signaling happens. While in this scenario, because JAZ is there, so there is a repression of the ABA signaling. So in this case, because there is a SKIP31, they can target JAZ protein and they can induce the ABA signaling pathway. So this is the overall conclusion and the working model. So in the JAZ actually repress, that's what we have shown, ABI5 activity. Here you can see the JAZ acts as a repressor for the ABA signaling pathway. They interact with the ABI5, so that's why there is a compromised seed maturation and seed vigor, because ABI5 cannot act for the signaling. This scenario also happens during the seed germination, because during seed germination there should not be any ABA signaling pathway. While in case of seed dormancy or maturation, there is a lot of SKIP31 protein in the during maturation stage. So they can target this JAZ protein and let them degrade to the 26S pathway, and then this, if the JAZ degradation, then there is a ABI5 is free, they can do their job and they express the downstream genes, which is important for seed maturation and vigor. So these are my lab groups. So actually whatever work I have today discussed is all done by the Vishal and some other scholars as well. and I also acknowledge the funding of Department of Biotechnology, Government of India, and our extensively NIPGR core grant. Thank you. Thank you for your attention.
Manoj, nice talk. So, what is the expression level of SKIP31 in recalcitrant seeds?
So
And
that,
can you express it there and make it?
yeah, that we have not checked, but in the other part of the story, like protein repairing enzyme PIMT, that is also another enzyme which is very essential to maintain vigor and viability for orthodox seed. So that particular enzyme is very less expressed in the recalcitrant seeds.
Right, basically, the question is that
Yeah.
can you manipulate in such a way that recalcitrant seed can also become long-lived?
Yeah, but in a plant system, no, transformation is very difficult. It's not like animal systems. So there are certain plants that can be transformable, like rice, variety of seeds, but recalcitrant seeds, they are not very susceptible for transformation. It's very difficult to transform them.
So let's thank
Okay.
Dr. Majee.
Okay, thank you, sir.
Let me